dear students. So in the last session we were discussing about the batch and continuous culture. So in today's session let's discuss about synchronous and diauxic culture. Synchronous cultures. Synchronous cultures are composed of populations of cells that are at the same stage of their life cycle. Synchronous growth of a bacterial population is that during which all bacterial cells of the population are physiologically identical and in the same stage of cell division cycle at a given time. Thus, the entire population is kept uniform with respect to growth and division. Synchronous growth helps studying particular stages of the cell division cycle and their interrelations. In most of the bacterial cultures, the stages of growth and cell division cycle are completely random, and thus it becomes difficult to understand the properties during the course of division cycle using such cultures. To overcome this problem, the microbiologists have developed synchronous culture technique to find synchronous growth of bacterial population. During scientific studies, it is not possible to analyze a single bacterial cell to obtain the information about growth behavior, that is, organization, differentiation, and macromolecular synthesis. Synchronous culture provides the entire cell crop in the same stage of growth. Measurement made on such culture is equivalent to the measurement made on individual cells. Synchronous cultures of bacteria can be obtained by a number of techniques. Two fundamentally different experimental approaches have been employed, and then the first one is a selection by size and age, and second one is a selection by induction technique. So now let's see about the selection by size and age. A population of cells is fractionated on the basis of size in this technique. The cells are filtered so that smallest cells pass through the filter. These small cells are the youngest and must go through their whole cycle before dividing. Alternatively, the largest cells which are ready to divide may be retained or retarded by a filter. These cells are then collected separately and used to obtain a synchronous culture. The most widely used method for obtaining synchronous culture is the Helmstedt-Cummings technique. And instead of filtration, density gradient centrifugation is also used to separate these cells. So now let's see in detail about the Helmstedt-Cummings technique. In this, unsynchronized bacterial culture is filtered through cellulose nitrate membrane filter. The loosely bound bacterial cells are washed from the filter, leaving some cells tightly associated with the filter. The filter is now inverted and fresh medium is allowed to flow through it. The new bacterial cells that are produced by cell division and are not tightly associated with the filter are washed into the effluent. Hence, all cells in the effluent are newly formed and are therefore at the same stage of growth and division cycle. And this effluent thus represents a synchronous culture. And in this image you can see the Helmstedt-Cummings technique of obtaining synchronous culture. The second method of obtaining synchronous culture is by the selection by induction technique. A synchronous culture is also obtained by the use of shock treatments. These include variation in temperature, starvation, exposure to light, drugs, and sublethal doses of radiation. A commonly used technique involves submitting a culture of microorganism to single or multiple changes in temperature. For example, an exponentially growing culture at 37 degrees Celsius is held for about 30 minutes at 20 degrees Celsius. So this culture from its favorable temperature is being exposed to another temperature, that is from 37 to 20 degrees Celsius. And the lower temperature returns to cell division, and during the interval of 30 minutes, all the cells mature to the point of fission. However, at 20 degrees Celsius, none of the cells will divide. On sudden return of the culture to 37 degrees Celsius, all the cells will divide synchronously. So it's like a culture that is growing in 37 degrees Celsius was exposed to 20 degrees Celsius for 30 minutes, and again it was exposed to 37 degrees Celsius, and at that stage all the cells will be dividing synchronously. By repeating this alteration of temperature, synchrony can be obtained in the culture for several generations. The technique based on selection are preferable to those based on induction, since induction is likely to introduce distortion in the physiological state of the cells. So there we come to the end of the synchronous growth. Now let's see what's happening in diauxic growth. Diauxic growth means double growth. It is caused by the presence of two sugars on a culture growth media, one of which is easier for the target bacterium to metabolize than the other. The preferred sugar is consumed first, which leads to rapid growth, followed by a lag phase. During the lag phase, the cellular machinery used to metabolize the second sugar is activated, and subsequently the second sugar is metabolized. Diauxic growth is a diphasic growth represented by two growth curves intervened by short lag phase produced by an organism, utilizing two different substrates, in this one of which will be glucose. When E. coli grows in a medium containing two substrates, that is glucose and lactose, it uses glucose preferentially until the glucose is exhausted. Then after a short lag phase, during which bacterium synthesizes the enzyme needed for the lactose use, growth will resume with lactose as a carbon source. If this diphasic growth of E. coli is plotted in respect to bacterial density against time, two growth curves follow one after the other, intervened by a short lag phase is obtained, and this is termed as diauxic or diphasic growth curve. And this graph represents the diauxic or the diphasic growth curve of E. coli, grown in a mixture of glucose and lactose. In this you can see that the glucose is first used and then it is followed by lactose, and you can see that there is a short lag phase in between. And in this lag phase, the bacterium synthesizes the enzyme that is needed for the use of lactose from the medium. So the graph explained about a simple example that involved the bacterium E. coli. The bacterium is grown on a growth media containing two types of sugars, one of which is easier to metabolize than the other, for example glucose and lactose, in which the glucose is easily metabolizable than the lactose. First, the bacterium will metabolize all the glucose and grow at a higher speed. Eventually, when all the glucose has been consumed, the bacterium will begin the process of expressing the genes in order to metabolize the lactose in the medium. This will only occur when all the glucose in the medium has been consumed, and for these reasons, diauxic growth occurs in multiple phases. The first phase is the fast growth phase, since the bacterium is consuming exclusively glucose and is capable of rapid growth. The second phase is a lag phase, while the genes used in lactose metabolism are expressed and observable cell growth stops there. This is followed by another growth phase, which is lower than the first because of the use of lactose as a primary energy source, and the final stage is the saturation phase. This type of diauxic growth can also occur when the bacterium in a closed batch culture consumes most of its nutrients and is entering the stationary phase. When new nutrients are suddenly added to the growth media, the bacterium enters a lag phase where it tries to ingest the food. Once the food starts being utilized, it enters a new log phase, showing a second peak on the growth curve.